This lecture is on cell cycle and mitosis. Listed here are your learning objectives. The cell cycle is just the way new cells are formed by division of a parent cell. So that one parent cell is going to grow and then divide and split everything up into two new cells. Reasons that your cells may need to do this: growth. So you're not the same size you were when you were a baby. You need more cells in order to fill in that amount of space. Development. So as you're growing and developing, your organs are changing and developing new aspects, or in the womb, development in the womb as well. Repairing and replacing cells. Repairing things from, say, a cut or cells that were infected with something, and replacing those with new cells. And reproduction, so making new organisms. The cell cycle consists of several phases. The G1, S, G2, and M phase. So this circle on the left-hand side shows all these different phases. Total cell cycle takes about 18 to 24 hours. That really depends on what kind of cell it is. It'll vary a little bit between the different types of cells. And this says once committed, meaning the cell has to decide that it's going to go down the pathway of making new cells. So it can stay in this G1 phase indefinitely. So it has to decide that it's going to move out of that G1 into the rest, and at that point it takes about 18 to 24 hours. Some cells can have much shorter cell cycles, so E. coli, only about 30 minutes. Most bacteria is this way, much shorter cell cycle than with mammalian cells. The first phase that we're going to talk about is G1. G1 is part of this interphase, which is this green circle all the way around. That's everything except for mitosis, when you're actually splitting the two cells apart. So G1 stands for GAP1. In this phase, you're going to synthesize proteins needed for replication, so DNA polymerases, helicase, histones, any proteins that you're going to need for that DNA. Synthesize more organelles, so you need enough mitochondria, for example, enough ER to pass on to the new cell. And the timing for this varies on cell type, so that can take anywhere from 8 to 10 hours. The synthesis phase is for DNA replication, so all of the DNA is going to be replicated. You need to make a whole new copy of DNA so that you have something to give to the new cell. And G2 phase, the gap phase, more growth. So your cytoplasm is increasing in volume. The cell's getting bigger because you want to make two normal size cells, not too many cells from the original. Expression of proteins needed for mitosis. So any of the proteins that are needed for that process of separating the DNA apart is going to be made in gap 2. And that takes from 4 to 6 hours. And finally, we have mitosis. Mitosis is the shortest part. It takes anywhere from 30 to 45 minutes, and it's the process of actually separating the chromosomes into two new cells and then dividing into those two new cells. Cytokinesis, it's not technically part of mitosis, but it's always kind of tagged on the end of mitosis, and it's the dividing of the cytoplasm and making those two new membranes. There are 5 stages in mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. And we'll go through each one and what happens in each. In prophase, this is the first phase after the GAP2 phase. So DNA has already been replicated, and you're ready to start separating the DNA into the two new cells. First thing that's going to happen are chromosomes are going to condense. So here's our diagram from back when we were talking about DNA packaging. And remember, up here, things are less packaged. You got your double helix, and then packaged with your histones. And as you go down this diagram, you get to the more condensed form, which is the kind of X shape that you remember seeing for chromosomes. So the idea is basically that you want them in a compact shape like this, because otherwise it's just kind of like a big ball of yarn, and that can get tangled very easily, and that can cause chromosomes to break, or they don't get sorted correctly into the two new cells. And remember, we talked about if you have extra chromosomes or not enough chromosomes in cells, that can cause really major problems. So that's the reason why they need to condense, to make them more compact, easier to pull apart and separate. Remember, this is after S phase, so you've got duplicate copies of everything. So recall that you've got 23 different types of chromosomes, and each type of chromosome, you've got 2 different copies, one from mom, one from dad. So when your cells go to divide, let's say this is chromosome 1 down here. This is chromosome 1 from mom or dad, this side of the X. And it's duplicated to make an identical copy, which is the other side of the X. And that's the duplicated form of that chromosome. So this X shape actually contains 2 copies of that chromosome. They're attached in the middle at the centromere. They actually don't cross, right? It looks kind of like an X, but it's actually 2 long pieces side by side, and then they're just connected with some proteins here at the middle called the centromere. The other thing that happens in prophase is that centrosomes start to form. So that's the microtubule networks that are going to help pull those chromosomes apart. Remember, microtubules are part of the cytoskeleton. They're big chains of protein that are going to help move these chromosomes apart. So they're going to form starting here. If you look down at this cartoony picture, starting here with the centrioles. The centrioles are the places of origination. That's where all the microtubules start from. So you can see these little lines coming out. Those are microtubules coming out from these centrioles. And those centrioles are going to start to move apart. They're going to be at opposite poles, so they can establish 2 different sides of the cell. So as they move apart, these spindles are forming. So these groups of microtubules, so if you look at this next picture with prophase, these microtubules here in between are forming the spindle. And that whole thing is called a centrosome. If you look at the microscope pictures up here at the top, the blue is the nucleus. The green are the microtubules. And you can see that right here at interphase, they're kind of together all at the same side. And now you can see these 2 bright light dots here. Those are the 2 centrioles, and they're going to start moving apart. You can also see here in the nucleus, notice how in prophase it looks kind of like almost like a brain. It's got all these little squiggly lines. Those are the chromosomes that you can actually see are condensed. You can't see that over here in interphase. It just looks like a solid blue circle. But you can see the chromosomes starting to condense here. Okay, prometaphase is kind of, I guess, in between prophase and metaphase, right? So it's kind of helps you remember which order that things go in. The nuclear membrane starts to fragment, meaning we want to get rid of that nucleus membrane, the membrane around the nucleus, so that we've got room to work with our chromosomes and actually divide those into 2 new cells. If they stay in the nucleus, it's going to be really hard to divide that nucleus into 2. So that's going to just almost kind of dissolve. Those lipids come apart. And then the microtubules are now going to connect to these chromosomes. So here we're just showing 4 sets of chromosomes. Remember, there's 1 copy on this side of this chromosome, and then the identical copy on the other side, held together by this little green piece here, the centromere. The microtubule is going to connect at that point. There's a protein complex that the green part's representing at the centromere called the kinetochore. And that protein complex is what those microtubules are going to connect to. And then there's going to be some kind of pushing and pulling as they line those chromosomes up where they want them. You can see in the microscope picture up here that the centrioles are now on opposite sides of the cell, and you can see the microtubules kind of starting to radiate out from it, and they're attaching to those chromosomes. You can see that the chromosomes are no longer contained by a membrane. During metaphase, the chromosomes are aligned at the middle of the cell. So those microtubules are connected at the kinetochore, and they're going to push and pull those chromosomes till they're lined up right across the middle. And it's actually a little more pushing than it is pulling exactly, but there's going to be some tension on either side from one side versus the other, and that's what's going to help them line up all in one place. When you look at the microscope picture, remember these chromosomes are really long, so it looks nice and neat in our cartoon picture, but these ends of the chromosomes that aren't held by the microtubules, they can kind of go wherever. So that's why it looks like little fingers kind of sticking out all over the middle of these cells. But this is what it looks like when they're lined up at the middle. Next is anaphase. This is the shortest phase. And during this phase, the sister chromatids are pulled apart. So we had them lined up at the middle. Now the chromatids are going to be pulled apart to opposite sides of the cell so that we can start forming two new cells. You can see that up here in the microscopy picture, with the green being the microtubules, and you can see the blue sections being pulled apart to different sides of the cell. So there are two types of motion when we're talking about these chromosomes being pulled apart. The first type is in this center panel, where the microtubules are actually pulling, and they're not truly pulling in the sense that we think of pulling. What's happening is this microtubule is getting shorter, so it's shortening from this end, and as it's shortening, then it's pulling that chromosome toward itself. The second type of motion is in the bottom panel, and that's the other microtubules, the ones that aren't attached to the chromosomes, and they push each other. So they're pushing each other so that these centrioles get further and further apart. So two sets of motion, both those combined help separate those chromosomes into two sides. And finally, we have telophase. It's coupled with cytokinesis. So after telophase, you're going to go straight into cytokinesis and separate that cytoplasm. But telophase you can kind of think of as the reverse of prophase. So the chromosomes are going to decondense. So they're going to go from that X shape and decondense so they can actually be used for transcription now. And that's what you want them for, right? You want that cell to be able to use those chromosomes to make RNA, to make proteins. So it needs to decondense in order to do that. And the nuclear envelope is going to reform. So we're going to have the envelope start to take place on both sides. So keep in mind that this is still one cell. It's still not divided yet. So we have to form two new nuclei to contain all of that DNA. And then finally we have cytokinesis. With animal cells, cytokinesis is going to happen. It's kind of like if you had a piece of Play-Doh and you just pinch it between your thumb and your forefinger until it pinches apart into two pieces. It occurs at the mid-zone, so between the spindles. So if you look at this picture on the right-hand side, all this white stuff are the spindles. They've already pulled apart chromosomes. They're already on the opposite sides, and you can see kind of this dark spot where there isn't any microtubules, and that's where it's going to start cleaving off in the middle. So we call this cleavage. The cleavage furrow, it pinches itself off, and that's made of some actin filaments, so other cytoskeleton components, actin, and they form kind of a ring right here around the middle, and it kind of acts like a belt, just squeezes off until you've got two new daughter cells. And that separates all the cytoplasm, all the organelles, everything else that's in the cell into those two new cells. All right, next we need to talk just briefly about cell cycle regulation. There's a lot that goes on with this, so we're just going to touch on it just very briefly. There are three goals in regulation. You need to make sure that each phase happens in the appropriate time and sequence. So you don't want anaphase to happen before metaphase. So each thing has to be carried out at the right time. Each phase has to be completed before the next point begins. So there are checkpoints to help make sure that DNA has been fully replicated before we start mitosis and that sort of thing. And that anaphase, so the chromosomes have been fully separated before we actually pinch off into two new cells in cytokinesis. And then the cell can respond to external signals that are telling it that it needs to make more cells. Okay, so here are some of the checkpoints and transition or restriction points. So G1 to S. So each of these red bars over here on the right-hand side are showing you a point at which the cell is going to check and make sure everything is going to is happening in the appropriate time and sequence. So G1 to S, this one right here. The cell may stop here indefinitely. And some cells do this because you don't need more. So if you don't need to make more heart cells right now, your cell is going to stop after that first growth phase, and it's just going to keep doing its normal functions as long as it needs to. There are other reasons that a cell might stop here, such as limiting nutrients. So if there isn't enough energy coming in, if you're starving, the cell might stop here and not doesn't want to waste energy dividing if it can't continue to support itself. Limiting space, so that's what I was just mentioning of if you've got enough heart cells and you don't need to make more, the cell's not going to go ahead and divide. It's going to wait until something is needed before it divides. And then inhibiting processes like protein synthesis. So things like viruses can stop protein synthesis from happening in cells. So if that's the case, your cell can tell there's something wrong. It's going to stop here because it can't make all the proteins it needs to go on with the process. All right, the next one is the G2 to M restriction point. So we're up here now. This can be influenced by cell size. So you don't need to keep growing and growing and growing and growing, right? There's a limit to the cell size. This one can also be stopped due to DNA damage or DNA replication. So maybe the S phase is taken a little too long. And we get up here and growth is still happening. You've made all the mitosis proteins, but there's still pieces of that chromosome that haven't been replicated. It's going to stop here and let the S phase catch up a little bit. Or perhaps there's DNA damage, so either through replication or DNA damage that was caused by UV light or a carcinogen or something like that, and the cell needs to fix it. It's going to pause here and let the cell try to fix it before it moves on. Now we'll talk about what happens if it can't fix that damage, but assuming that it just needs a little extra time, it'll go ahead and fix the damage and then it'll continue on. And then the last part here is metaphase to anaphase. So this is during mitosis. Remember, metaphase, the chromosomes are lined up across the middle, and anaphase is when they pull them apart. So the thing that would make the cell stop here are chromosome attachments to the spindle. So if those microtubules don't attach to the chromosomes correctly, or maybe only one side attaches but the other doesn't, if that cell goes on through anaphase, it's going to pull those chromosomes apart and they're not going to separate appropriately. So the cell wants to try to prevent that. So it might pause here. Okay, I'm just going to tell you about one of the checkpoints that might occur. This is the DNA damage checkpoint. It uses a protein that's very, very famous called P53. And this protein has the ability to activate two different responses depending on how bad that damage is. So what initially happens, up here we got a little red kind of star thing showing you that there's some kind of DNA damage that happens. So we already learned that if DNA damage happens, the cell's going to try to fix it somehow. But first it needs to pause the cell cycle so it has time to do that. So initially that's going to activate some proteins. These are proteins called ATM, ATR. You don't need to remember those names, but just know these proteins are ones that are going to recognize, hey, there's damage here, and we need to signal to the rest of the cell that we need to pause and not go through the rest of the cell cycle just yet. So it's going to signal to some checkpoint kinases. Remember, kinase phosphorylates another protein. So it's going to phosphorylate this protein P53. It's shown here in purple. P53 is a transcription factor, so once it's phosphorylated, it's going to travel to the nucleus, go in the nucleus, and it's going to activate transcription of some genes. So initially, this P53 is going to activate genes that are going to lead to cell cycle arrest. So their primary job is to make the cell cycle pause. If that damage is too bad, it's very severe, it's not being fixed, that P53 can activate another set of genes. In that case, that set of genes actually leads to apoptosis or cell death. So the cell at that point has said, it's too bad, it can't be fixed, we need to get rid of this cell so it doesn't cause damage to any of the other cells around it. That leads us to talking about programmed cell death or apoptosis, or you might hear it pronounced apoptosis, depending on who you're talking to. And this is a cell-induced death, so that the cell itself is inducing its own death. It's very programmed, it's very scheduled, and it's doing that so it's going to prevent damage to other cells around the dying cell. So here I'm showing you two different types of cell death. So we got apoptosis here on the right, where it's kind of breaking itself down. So you see different pieces kind of breaking off from each other. And those will just go to a phagosome or some other type of cell that's going to come engulf those and just get rid of them. Versus necrosis, which just the cell ends up bursting, and then all the stuff inside the cell can get out and affect all the other cells around it, and it can damage those. So you want to try to prevent this. That causes a lot more damage. This limits the damage a lot. Okay, a lot of different reasons apoptosis is used. So active during development. So this is that idea that we talked about, that initially during development your fingers are webbed and your toes are webbed, and the cells between them go through this apoptosis in order to separate them into your fingers. It might be after problems with the cell cycle like we just talked about. So P53 gets activated, damage is too bad, we got to get rid of this cell. It might be some type of infection. So viruses can activate apoptosis, not intentionally, but the virus itself, when it gets in and starts infecting and making more of itself, can activate the apoptosis pathway to try to limit infection, get rid of that virus before it spreads to other cells. And then it's active with aging cells. So any cells, for example, we talked about skin cells, intestinal cells. These cells don't hang around too long. You replace your skin very quickly. Those cells need to be replaced. So the old cells will go through apoptosis and be replaced with new ones. There are several different events that happen. We already talked about membrane lipids that get inverted, so the phosphatidylserine flipping to the other side of the membrane, and that's going to help with that the process of blebbing that we'll talk about in a minute. DNA starts to get fragmented. Remember, we're just in a very organized way breaking down all the parts of these cells. So the DNA gets cut up into shorter pieces that can then be recycled or put out in the waste much easier. So there are nucleases that are in charge of doing that, and they're cut into segments of 180 base pairs or multiples of that. Reason for that is there's histones in those in that DNA. So those endonucleases can only cut between the histones, and it's about 180 base pairs from each nucleosome to the next. So that's why it comes off in those types of segments. You can see that if you look on a gel. So over here, in the first lane, you got the DNA markers. So this is known sizes of DNA. And then you've got 1, 2, 3, 4, 5 hours over which they watched this happen. So here you start out with all your DNAs. The size is pretty big, and you can see the cleavage of that DNA start to happen, or the fragmentation. And you see it's a very regular pattern, little ladders here over the course of several hours. Blebbing occurs with the membrane. So we start out with our cell here. It's starting to break down. You're going to lose a lot of the water from that cell, from the cytoplasm, and then that membrane is going to start breaking off into small little vesicles, and that'll be taken up by a phagocyte, white blood cell, and just broken down, taken care of. We'll get rid of that piece of cell. Here down at the bottom, you can see the cells that are blebbing. You can see these little pieces of membrane that are coming off of that cell. This process can be activated by something called death signals. These are signaling molecules that are sent to the cell to activate it, to get rid of it for, if it's damaged, if it's infected, those sorts of things. The cell can use it to signal to itself, so it can be autocrine signaling, where it's signaling to itself to die. Or it could be lack of survival factors. So there's not enough nutrients, we're starving, it's going to start to die. Both these things have a similar result no matter which way it's activated, and that's the activation of caspases. Caspases are enzymes, and specifically, here's our definition, they're cysteine aspartic acid proteases. So a protease is an enzyme that's going to digest proteins. So we already talked about the membranes being taken care of with the blebbing, the DNA is being taken care of by fragmenting. Now we have to get rid of the proteins as well. So use proteases to do that. It's going to go about digesting all the proteins in the cell. There are two types of caspases. One's called initiator and one's called executioner. And they exist in the cell all the time. They're called procaspases, but they must be cleaved to be activated. So the initiator is the first one that's activated, and then it's going to cleave the other caspases, meaning it's going to cut a little piece off, and when it does that, it activates that protein, and then it can cleave, the executioner caspases can cleave all the other proteins found in the cell.